Good afternoon, all of you. This is Dr. Thenmozhi, Head of the Department of Zoology, St.Joseph's College of Arts and Science, Cuddalore. I am going to teach the topic about immunoglobulins, its structure and functions today. First of all, I should say what is immunoglobulin. Immunoglobulin is nothing but antibodies. Other name is antibodies. They are made up of glycogen and protein. It is a glycoprotein molecule produced by our plasma cells, that is our white blood cells. Major of them are produced by the white blood cells. They act as a critical part of the immune response by specifically recognizing and binding to a particular antigen such as bacteria, viruses, or aiding in their destruction. This is a diagram that shows the structure of an immunoglobulin. Many immunoglobulin have different types of structures, even though this is the basic structure of an immunoglobulin, which is having a 4-chain structure, that is 2 light chains and 2 heavy chains, and it is Y-shaped, in which the anterior part is the variable, having the variable domain with the antigen binding site. Then it is also having the 2 light chains and 2 heavy chains are there, in which one of the part is having domain, variable domain, that is it can changes its shape, then it is called as variable domain, that is yellowish in color, and the which one is a constant domain, otherwise it won't changes its shape, that is blue in color. Okay, this constant domain, they may not changes its shape. Then fab is a fragmentation antigen binding site, and FC is another region, that is the Y, the vertical region of the Y shape, that is called as FC, fragmentation crystallizable site, in which between this there is a disulfide bonds, that is non-covalent disulfide bond is there, which is flexible in nature. The basic structure of an immunoglobulin, it consisting of the basic unit, 4-chain structure only. Different immunoglobulins can differ structurally, but they are all built from the same basic pattern. Heavy and light chains. What are heavy and light chains in an immunoglobulin? All the immunoglobulins have a 4-chain structure, as I said earlier, as their basic unit. They are composed of 2 identical light chains and 2 identical heavy chains. In between this interchain disulfide bond is present. The heavy and light chains and the 2 heavy chains are held together by interchain disulfide bonds and by non-covalent interactions. The number of interchain disulfide bonds varies among different immunoglobulin molecules. Intrachain disulfide bonds. Within each of the polypeptide chain, there are also intrachain disulfide bond is present. What is variable domain and constant domain? Each chains are having 2 domains, variable domain and a constant domain. When an amino acid sequences of many different heavy chains and light chains were compared, it has become clear that both the heavy and light chains could be divided into 2 regions based on variability in the amino acid sequences. It is based on the amino acid sequences only. That is, the light chain, that is represented as VL. VL means variable region in the light chain, that is composed of 110 amino acids, and constant light chain, that is composed of 330 to 440 amino acids. In the same way, heavy chain also consisting of 2 domains, that is variable domain and a constant domain. In variable domain of heavy chain, it also consisting of 110 amino acids, whereas in the light chain, also we have the same structure, that is with 110 amino acid structure. And in the constant region of the heavy chain is also the same, 330 to 440 amino acid. It is made up of 330 to 440 amino acids. That is constant region of the heavy chain. Hinge region. What is hinge region? This is the region at which the arms of the antibody molecules forms a Y. It is called hinge region because there is some flexibility in the molecule at this point. Domains. On seeing the three-dimensional image of immunoglobulin molecule, that it shows that it is folded into a globular region. Globular means globular proteins only, each of which contain an intra-chain disulfide bond. These regions are called domains. The light chain domain, heavy chain domain, as we have seen earlier. In the light chain domain, we can see two regions, that is variable light chain, constant light chain. Likewise, in the heavy chain, we have two domains, that is variable heavy chain and constant heavy chain. Constant heavy chain will be polymorphous, that is exist in four forms, that is CH1, CH2, CH3 and even CH4. Up to CH4 we can see in the heavy chain domains. Carbohydrates are attached to the CH2 domain, that is constant heavy chain 2 in the form of 2 structure domain. We can see it is made up of carbohydrate. However, in some cases carbohydrate may also be attached to other locations also. Now we can see immunoglobulin classes. Immunoglobulin can be divided into 5 different classes. It is based on the differences in the amino acid sequences in the constant region of the heavy chains only. That is immunoglobulin G, that is gamma heavy chain. Immunoglobulin M, mu heavy chain. Immunoglobulin A, alpha heavy chain. Immunoglobulin D, delta heavy chain. Immunoglobulin E, that is epsilon heavy chain. So these 5 classes of immunoglobulin can be remembered. So it is also having some subclasses. The subclasses of immunoglobulins, again the classes can be divided into small differences because of the small differences in the amino acid sequences of the constant region of the heavy chain. All immunoglobulins with the subclass will have very similar heavy chain constant region amino acid sequences. That is, in immunoglobulin G subclasses you can see 4 types, immunoglobulin subtypes, 4 subtypes. Immunoglobulin G1, immunoglobulin G2, immunoglobulin G3 and immunoglobulin G4. That is gamma 1 heavy chain, gamma 2 heavy chain, gamma 3 heavy chain and gamma 4 heavy chains. Immunoglobulin A subclasses, there are 2 subclasses under immunoglobulin A, that is immunoglobulin A alpha 1 heavy chain and immunoglobulin A2, that is alpha 2 heavy chains. Immunoglobulin types, that is can be again classified into light chain, how many type of amino acid sequences it depends. Light chain types are based on differences in the amino acid, as I said already. That is 2 types of light chains we can see. It is already we have seen heavy chain only, now it is light chain, kappa light chain and lambda light chain. Immunoglobulin light chain subtypes, you can see lambda 1, lambda 2, lambda 3 and lambda 4. Now we can see the general functions of immunoglobulin. Its first function is antigen binding. Of course, antigen binding with the antigen is the first and foremost function of immunoglobulin. Immunoglobulin bind specifically to one or few closely related antigens sometimes. Each immunoglobulin actually binds to a specific antigenic determinant site. Antigen binding by antibodies is the primary function of the antibodies because it can result in the protection of the host. What is the valency of the antibody? What do you mean by valency? Valency is nothing but the number of binding sites in an immunoglobulin. So the valency of the antibody refers to the number of antigenic determinants, that is binding site, nothing but binding site. Antigenic determinants that an individual antibody molecule can bind. So the valency of an antibody should be depends upon the number of binding site which is present in it. But it is a rule that the valency of all the antibodies must at least 2. Effector function. This is another one function of immunoglobulin, that is fixation of the complement. This is another one important function of it, because for the lysis of the antigen, antigenic cells should be done by these immunoglobulins only, by releasing biologically active molecules in the blood serum. Binding to various cell types like phagocytic cells, lymphocytes, platelets, mast cells, and basophils. They also have the receptors that bind with the immunoglobulins. Some immunoglobulins also bind to receptor on placental trophoblast. That is nothing but special kind of cells, immunological cells produced by the placenta. So these placental trophoblast for transferring the immunoglobulin across the placenta. So the transferred maternal bodies provide immunity to the fetus and the newborn. So thank you, students.